So let us see the mismatch repair system in the human beings. So Paul Modric and co-workers have reconstituted the human mismatch repair system in vitro. A strand break on either side of the mismatch is sufficient to start this repair. So if a mismatch is in the DNA and only a small break on either side of the mismatch is present, the mismatch repair system will start. Purified human mutase alpha, mutal alpha, exo 1. Exo 1 is a 5' to 3' exonuclease. RPA, PCNA, RFC, and DNA polymerase delta are required for bidirectional repair. So you see a lot of enzymes and protein factors are required in human beings for this mismatch repair, and these include some enzymes as well as some protein factors. Mutase alpha, exo 1 and RPA are adequate to excise a mismatch when the nick is on the 5' side of the mismatch. So these three are required when the nick is on the 5' side of the mismatch. So this is you can see this is a strand, and here in the center, if the mismatch is here and nick is on the 5' side, then these three, that is mutase alpha, exo 1 and RPA are required, while mutal alpha, PCNA and RFC are also required when the nick is on the 3' side of the mismatch. So in addition to above three factors, these three more factors are required when this nick is on the 3' side of the mismatch. The observation that the mismatch repair system can degrade newly synthesized strands with a nick on the 3' side of the mismatch was very puzzling, because the exo 1 degrades DNA in a 5' to 3' direction. So it starts degradation from 5' to 3 direction. But when the nick is present on the 3' direction, how this enzyme will degrade from 3' to 5'? So this was a very puzzling. Modric and co-workers solved the puzzle by demonstrating that mutal alpha is a latent endonuclease that is activated in a mismatch. Once activated, this mutal alpha preferentially makes incisions in the strand that already has a nick, that is the discontinuous strand during replication. So you know, during replication, this discontinuous strand is a newly synthesized strand, and obviously this strand would have a nick. So this mutase alpha will make an incision in this strand. The endonuclease activity appears to require an amino acid motif present in the PMS2, but not the MLH subunit. So it doesn't require MLH1 subunit, rather it requires an amino acid motif. In the human mismatch repair pathway, mutase alpha, PCNA and RFC, they cooperate to activate the mutant mutal alpha endonuclease, which was a latent endonuclease. This mutal alpha endonuclease then nicks the discontinuous strand of the DNA duplex on both the 5' side and 3' sides of the mismatch. So once it is activated, it introduces two nicks on both sides, that is 5' and 3' of the mismatch. When the original nick is on the 3' side of the mismatch, mut L alpha incision produce a new 5' terminus on the far side of the mismatch that can serve as an entry site for mut S alpha activated exo 1. So if the initial nick is on the 3' side, a new nick is introduced on the far side of the strand, that is on the 5' side. This activated exo 1 then removes the mismatch using its 5' to 3' exonuclease activity. So this mismatch is then removed by hydrolyzing this discontinuous strand. RPA stimulates exo 1 activity in the presence of mut S alpha as long as the mismatch is present. So if the mismatch is present, this RPA stimulates the activity of exo 1. Then exo 1 start hydrolyzing this strand. Once the mismatch has been removed, RPA inhibits the exonuclease. So when this mismatch is removed, this exonuclease is inactivated, probably by displacing exo 1 from the DNA. So exo 1 is displaced from the DNA. So in this way, this RPA inhibits the exonuclease. So here in this picture, you can see these two sides, these two pictures, and this picture demonstrates the 5' directed nick repair, and this picture describes the when the nick is on the 3' side. So here you can see this is mut S alpha, and this is mut L alpha, and this is RFC and PCNA. So this nick is present on the 5' side of mismatch. Actually, mismatch is here. So the nick is on 5' side. So this is new nick on the 3' side, and then in the presence of mut S alpha, mut L alpha, exo 1 and RPA, this strand which was present here and which contained the mismatch, this has been hydrolyzed. Similarly, here you can see this is a 3' directed mismatch repair. So mismatch is present here and a nick is present on the 3' side here. So in this way, a new nick is introduced on the 5' side, and then this is hydrolyzed from 5' to 3' direction in the presence of mut S alpha, mut L alpha, exo 1 and RPA. So both these reactions, this reaction and this reaction, similarly here, this reaction and this reaction, these are coupled with the production of energy from ATP hydrolysis. So this is a brief of the human mismatch repair system. So individuals with a non-functional mismatch repair system due to defective mut S alpha or mut L alpha suffer from non-polyposis colon cancer, HNPCC, human non-polyposis colon cancer, and autosomal recessive syndrome. So this will happen if mut S alpha or mut L alpha are non-functional in the human beings.